Yes, myself Yojana Patil and my presentation topic is chromosome rearrangement and cancer. Chromosome rearrangement and cancer. So here is the content that we are going to see in above presentation, which is what is a cancer, what is chromosomal rearrangement, chromosome inversion, deletion, duplication, translocation, and the disease caused by chromosomal rearrangement, which is nothing but an Philadelphia chromosomes and an Burkitt's lymphoma. So first, let's see what is an cancer. So cancer is nothing but a disease which in which some of the body cells divides rapidly, abnormally, and uncontrollably, and they can invade the nearby tissues and spread to another part of the and also spread to another parts of the body. Now let's see what is a chromosomal rearrangement. So chromosomal rearrangement is refers to an changes in structure or the number of chromosomes that occurs in a cancer cells. Such changes may occur in and many ways like chromosome deletion, chromosome inversion, chromosome duplication, and chromosome translocation. These rearrangements may lead to an activation of an oncogene and disruption of an normal cells and a progression of cancer. Chromosomal rearrangement can be caused by exposure to UV radiation. So now let's see what is a chromosome inversion. So chromosome inversion is occurs when an a portion of an chromosome breaks off and it reattaches within the same chromosome, but in an reverse orientation, and this can results in an uh, alteration of gene regulation, and this can lead to cancer. As we can see in the diagram, uh, there is an inversion. So now let's see what is an chromosome deletion. The chromosome deletion occurs when a segment of chromosome is lost. During this, the loss of an important tumor suppressor genes may occur, which can lead to the formation of an cancer. Then chromosome duplication. So chromosome duplication is a type of chromosome rearrangement where in which a segment when a segment or a portion of chromosome is copied, and this can be results in a overproduction of oncogene, which can lead to cancer formation. So chromosome translocation occurs when a chromosome break off and the fragmented piece reattaches to the different chromosomes, and the detection of this chromosomal translocation can be important for the diagnosis of certain genetic diseases and disorders, and this can be result in formation of abnormal gene production, and that can cause cancer. Now let's see the types of an certain rearrangement of human cancers which are associated with the chromosome rearrangement. So first one is an Philadelphia chromosomes, and second one is an Burkitt's lymphoma. This Philadelphia chromosome is an human cancers which is associated with the chromosome rearrangement. For example, chronic myelogenous leukemia, which is associated with the aberration of an chromosome 22, and this Philadelphia chromosome is one of the important translocation between the chromosomes that generates the oncogenes. The second one is an Burkitt's lymphoma. It is an example of an white blood cell cancer, and it is associated with the reciprocal translocation. So here is an diagrammatical view of an Philadelphia chromosomes, where we can see uh, that the tip of an long arm of the chromosome 9, which has been joined to the body of the chromosome 22, and the portion of an long arm of chromosome 22 has been joined to the body of chromosome 9. In this translocation, the breakpoint on the chromosome 9 is within an CABL oncogene, and this encodes the tyrosine kinase. The breakpoint of an chromosome 22 is within an gene BCR. is within an gene BCR. Through this translocation, the BCR and CABL genes have been physically joined, and they creates a fusion which which has an amino terminals of BCR protein and carboxyl terminals of an CABL proteins. The mechanism of this involves the tyrosine kinase activity of an CABL protein, which is tightly controlled in normal cells, but it is degraded in the cell that produce the fusion polypeptides. The CABL protein is constitutively activated by this CABL protein is activated by an BCR and CABL gene fusion, and this fusion is therefore a dominant activator of CABL tyrosine kinase. And the deregulation of the CABL tyrosine kinase leads to the abnormal phosphorylation of the other protein. In this phosphorylated state, this protein causes cell to grow and divide uncontrollably, so it can cause cancer. So the second one is an Burkitt's lymphoma, which I said that it is an white blood cancers which is associated with the reciprocal translocation. In this the translocation invariably involves the chromosome 8 and the one of the three chromosomes 2, 14, and 22 that carries a gene which forms a immunoglobulins antibodies. In this the this translocation involve the chromosome 8 and 14 which are the most common. In this translocation, the CMYC oncogene on the chromosome 8 is juxtaposed to the gene for immunoglobulin heavy chain on a chromosome 14. This rearrangement results in an overexpression of CMYC oncogene in cell that produces the immunoglobulin heavy chains, that is the B cell of an immune system. This CMYC gene encodes the transcription factors that activates the genes which are involved in promoting cell division. Consequently, the overexpression of this CMYC genes occurs uh, in cell which carries an immunoglobulin uh, immunoglobulin heavy chains, and the CMYC fusion created by an 814 chromosomes, and this translocation causes those cells to become the cancerous. Here is a diagrammatical view of an Burkitt's lymphoma, that how is causes and how this translocation takes place in humans. These are the reference which I referred, and thank you. Yes, sir. Should I stop presenting? Okay, sir. Initiate the slide. Okay, sir. Which one, sir? Okay. Wait a minute, sir. Full form of CABL. Abelson Murine leukemia, I guess. So, sorry, sir. हाँ 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 chromosome. Uh, don't know, sir. Yes, sir. BCR is a breakpoint cluster region. हाँ sir. No sir. No, no, wait sir. नहीं सर, ब्रेक पॉइंट क्लस्टर रीजन ही है। हाँ सर। Tyrosine kinase. Sir, it encodes CABL oncogene. टायरोसिन काइनेज अगर कंटिन्यूअसली फॉस्फोराइलेट हुआ तो। हाँ सर, बना है। हाँ। हाँ। ओके सर।
सर मैं दीप्ति। Sir, is my screen visible? हाँ सर, घुमाया है अभी। Good evening, sir. Myself Dipti Khatpe, and my today's presentation topic is proto-oncology. These are the contents of my slide, which we are going to see. Uh, See first introduction. There are four classes of normal regulatory genes. These are the principal target of genetic damage. First is growth promoting genes, proto-oncogene. Second one is growth inhibiting tumor suppressor gene. Third is genes that regulated program cell death or apoptosis. Fourth one is DNA repair gene. Here we will see only first proto-oncogene. Proto-oncogenes have multiple roles, but all participate in signaling pathways that drive proliferation. Uh, Proto-oncogenes are normal genes involved in cell growth and division. Proto-oncogenes are genes that encode different kinds of proteins such as beta growth factor receptors, transcription factors, and or signal transduction proteins. So basically, proto-oncogenes are a normal genes present in our body. When these genes are ran into mutation or chromosomal rearrangement and gene amplification, we get the transformation of proto-oncogenes into oncogenes. These oncogenes are abnormal genes and this causes cancer. What are the some types of proto-oncogenes? There are four types basically. We see one by one. First is tyrosine kinase, tyrosine protein kinase like SRC and ABL. SRC is the regulation of embryonic development and cell growth, but when it is mutated or overexpressed, it causes colon cancer. Or second one is ABL gene. It aids in cell differentiation, cell division, and cell adhesion and stress response. It causes chronic myeloid leukemia or Philadelphia chromosome formation. Second is kinase related protein, that is RAF. RAF is the kinase related protein. It is the part of ERK pathway. When ERK pathway is continuously, that leads to the uncontrolled cell division and this causes the cancer. Third one is growth factor is SIS. This encodes beta chain of PDGF. PDGF. This causes neoplastic transformation of fibroblasts. And last one is nuclear protein like cellular oncogene, also called as C oncogene, normal oncogene, also called as N oncogene, MYB, and FOS. All these genes have capability to become oncogene once mutated and amplified. Like chromosomal translocation of CMYC with IgA gene, which leads to the Burkitt's lymphoma. And next, proto-oncogene. How we see how proto-oncogene are changed into oncogenes. When proto-oncogenes are changed into oncogenes, the diet control can be lost and unregulated cell proliferation can take place. First one is mutation, second is gene amplification, and third one is chromosomal rearrangement. We see only one by one. First is mutation. Uh, Mutation is also called as a deletion or second is regulatory mutation. In this point mutation, we get abnormal non-functional protein, whereas in regulatory mutation, we get the normal protein, but it is always overexpressed. Point mutation in RAS gene that causes cancer. In regulatory mutation, which mostly happens in enhances sequences or DNA or mutated. Second is gene amplification. A normal gene is expressed in amplified, or its expression is amplified, and from that second one is gene amplification. It's when normal gene is expressed or expression is amplified, and from that normal protein is overexpressed, which is ultimately leads to the formation of oncogene. And the last and biggest one is chromosomal rearrangement. The, the last and biggest transformation way of transforms proto-oncogene to oncogene is chromosomal rearrangement. These are of two types. First is chromosomal rearrangements in regulatory chromosomes, and second is the chromosome rearrangement in protein coding sequences that causes fusion of two genes. In rearrangement of regulatory sequences, the normal protein is overexpressed, while in the rearrangement of coding sequences, there is a formation of hyperactive fusion protein like BCR and ABL rearrangement. Then next we see normal RAS and protein RAS. Normal RAS and protein and mutant transprotein. In normal RAS protein, in this diagram, we see extracellular signal enters the cell through plasma membrane. In cytoplasm, inactive and active RAS protein is present. Inactive RAS protein is always bind with GDP, and in active RAS protein is bind with GTP. In cytoplasm, RAS protein is activated by phosphorylation of bound GDP and inactivated by dephosphorylation of bound GTP to stop the signaling. The active RAS protein is transduced this signal to the nucleus. This signal regulates the transcription of genes involved in cell division. There is the formation of DNA and RNA, and cell division occurs in a controlled fashion, and cell growth in normal RAS protein. In mutant RAS protein is unregulated. Extracellular signal has no influence on the state of the mutant RAS protein. In cytoplasm, mutant RAS protein is already in active state. Then it transmits a constituted signal to the nucleus. This signal causes gene involved in cell division to be transcribed in inappropriate manner. There are formation of RNA and cell division occurs in an uncontrolled fashion and causes a cancer. How normal cells are changes into uh, oncogenes. Proto-oncogenes are changed into oncogenes by point mutation and also called as a base pair substitution. Coding region or in the controlling sequence increase the activity of the gene product or the expression of the gene protein product. Example is the RAS mutation. Second one is the deletion. Changes in the amount or activity of the encoded growth factor, growth stimulatory protein, causing unprogrammed activation of some cell proliferation genes. Example is MYC genes can arise from its proto-oncogene by deletion. And second is last one is the gene amplification. Increased number of copies of the gene. Some tumors have multiple copies of proto-oncogenes. Let's see the function of proto-oncogenes. Proto-oncogenes are a group of typical genes in a cell. They contain the necessary information for our body uh, to make the protein is responsible for stimulating cell division, which makes cell growth possible, inhibiting cell differentiation, which is when cells change in their function, preventing apoptosis, also known as cell death. These processes are all essential for cell to maintain healthy tissues and organs in our body. Let's the example of proto-oncogenes. About 100 to about 100 proto-oncogenes have been identified. This uh, falls into several distinct classes based on the DNA sequences, similarities, and the similarities in amino acid sequences of the protein product. Over these 40 different types of proto-oncogenes have been discovered in our body. First is growth factors, proteins such that P, ONC, or C proto-oncogenes. These alter growth factors give next in protein kinase such that SRC, such that next is protein kinase such that SRC, and last is membrane-associated G protein, and activated by surface receptor, such that RAS protein which is encoded by the RAS gene. RAS it encodes an intracellular RAS it encodes an intracellular signal transduction protein. RAS is one of the on and off switch pathway. ACR2. This gene, these gene makes protein receptors that are involved in the growth and division of cells in the breast. Many people with breast cancer have a gene amplification mutation in their HER2 gene. MYC, these gene are is associated with the type of cancer called as Burkitt's lymphoma. Burkitt's lymphoma is a secretion of gene that enhances the gene function and gets moved near the MYC proton gene. And the last is cyclin D. Cyclin D, its normal job is to deactivate the protein called the RB tumor suppressor. When cyclin D mutates, it can no longer accomplish this, causing uncontrollable cell growth. These are my references. Thank you. RAS. Okay. Virginia. SRC. I, dominant, sir. Because
सर मैं
Yes, sir. Yes, sir. My name is Bhagyeshri Manmode. I am from TY BSc class. My roll number is 227319. My presentation topic is identifying genes by complementation mutation. Complementation, it is a test. It is also called cis-trans test. Uh, It determines whether two independently isolated mutants with the same phenotype have mutations in the same or different genes, like crossing two mutants. Uh, it discovered by American geneticists. Uh, Edward B. Lewis to study genes in Drosophila. In this in this test, two mutants resulting in the same phenotype are crossed, and the phenotype of progeny is observed. If the two mutations involved are in different genes, then the progeny will be wild type or mutant heterozygotes for each of the two genes involved. So here two diagram of mutations in different genes is a diagram, and mutations in same gene is B diagram. So A diagram is show that result is last wild type for phenotype genes we got, and B diagram shows that no complementation here, so mutant phenotype we got. So how the identifying genes by complementation mutation? So it is possible to clone genes by complementation of well-defined mutations. In brief, the expression of the wild-type gene introduced into the cell by transformation, overcoming the defect of mutant form of gene in genome. To clone a yeast, means Saccharomyces cerevisiae, gene by complementation, first a genomic library is made up of DNA fragments from the wild-type yeast strain. Then the library is used to transform a host yeast strain carrying two mutations, mutations to allow transformants to be selected, uh, URA3, for example, which gives a uracil growth requirement, and a mutation in the gene for which the wild-type gene clone is sought. So here is a diagram of with example of cloning a gene by complementation of mutation. The cloning of the yeast by ARG1 gene, arginine. So here in first step, a genomic library is made up using DNA from wild type RG1 yeast strain, and Ura A, Ura3 is a selectable marker. Then then when a population of Ura3 RG1 yeast cells is transformed with the genomic library prepared in the shuttle vector, then some cells receive plasmids containing the normal RG1 gene for the arginine biosynthesis enzyme. The plasmid's RG1 gene is expressed, enabling the cell grow on minimal medium, that is, in the absence of arginine, despite the presence of defective RG1 gene in the cell's genome. So then uh, fifth step is chromosomal RG1 makes defective enzyme, and last sixth one is complementation occurs because RG1 in vector produces functional enzyme. So the plasmid is then isolated from the cells and the clone gene is characterized. So, conclusion, complementation occurs when two mutations together result in a wild-type phenotype. Non-complementation occurs when two mutations together result in mutant phenotype. It is simple and fundamental assay in genetics used to assign a mutation to a gene. The number of units of genes is determined by complementation test. Given a set of mutations expressing the same mutant phenotype, the two mutants are combined and the phenotype is determined. So here is a references. cis means same chromosome carried on same chromosomes. The so, same chromosomes carry out होते और वो configuration so no, same chromosomes for same chromosomes तो carried out होते हैं और फिर configuration होता है तो तभी cis configuration बोलते हैं उसको नहीं cis configuration तो trans only one mutation happened there.
Yeah.
Thank you,
Hello,
sir.
sir.
God.
Yes, sir. सर दिख रहा है. Good, good evening, sir. Myself, Sakshi Jadhav. So my topic is genomic library. In humans, approximately 25,000 genes exist among the 3 billion base pairs of DNA in the genome. To study any one of these genes, a researcher first isolates it from all of the other genes in organism DNA. One isolation method has a relatively long history and involves the construction of DNA library. When a gene is identified and copied, it is said to have been cloned. So there are two types of DNA library, genomic library and cDNA library. So we are going to see what is genomic library. Contains gene fragment representing entire genome of an organism created using molecular cloning. Genome size is expressed in terms of number of base pairs. The size of genome in different species are variable. There is a distinct difference in the gene of prokaryotics and eukaryotics. In prokaryotic structures, genes coding for proteins are continuous, while in eukaryotes, the coding region exons are separated by non-coding regions. Therefore, the construction of gene libraries for eukaryotics is more complicated. Steps of genomic library construction: isolation of DNA from cells, second, digestion into small fragments, then introduction into suitable vectors, insertion into bacteria, and then DNA isolation collection of genomic DNA library. So, what are the uses of genomic library? Researchers can explore the genome of an organism to learn more about genomic structure and function. They can map the genome, identify the location of specific gene, helps to develop tests which can be used to locate genetics variation, include mutation. Useful in recombinant DNA technology helps to genetically modify organism and produce clone of desired types. Genomic library construction is the first step in DNA sequencing project. Genomic library helps in identification of the novel pharmaceutically important gene. Advantage of genomic library. So, libraries derived from eukaryotic organisms are critical for studying the genome sequence of particular gene of interest. It is useful for prokaryotics with small genome to identify a clone encoding a specific gene of interest. It helps researchers to explore more about organism genomic structure and function. It is also used to study genomic mutation genetic mutation. Pharmacologically important genes can also be identified by this method. So the disadvantage of genomic library, it requires sophisticated software and vast amount of computing power. Also, the process is prone to error. Eukaryotic genome library with large genomes contains many DNA that do not code for proteins as well as non-coding DNA like repetitive DNA and regulatory regions, making them less than ideal. Thank you, sir. Sir, again. Um, सर जीनोमिक में लाइक हेलो सर हेलो सो द जी genomic library comprise the large DNA fragments then cDNA library I don't know ah hello protein coding the exon.
Yes, sir.
Hello, sir. सर असाइनमेंट कब तक सबमिट करना है? सर लेट नाइट हो जाएगा चलेगा? ओके ओके
Yes, sir. Yeah, so shall I start? Hello, my name is Ashwati, and my today's topic is cancer and the cell cycle. So in my presentation, the table of contents are cancer, the cell cycle, and relationship of cancer and cell cycle. So first, when we talk about cancer, cancer is one of the leading cause of death worldwide. The cause of cancer is complex issue, but well-known risk factors are alcohol, tobacco abuse, and UV radiations. So this diagram explains how a normal cell, if we don't provide it a growth factor, it would, if we don't provide a growth factor, it would not divide. But when a cancer cell, if we don't provide growth factors, or if we provide growth factors, or if we don't provide growth factors, it is going to divide because it is a property of cancer. And cancer is basically an uncontrolled division of abnormal cells. There are four major types of cancers, which is carcinomas, sarcomas, leukemia, and lymphoma. In an adult, the many different types of cells of the body proliferate only in a controlled way. Occasionally, dividing and differentiating cells deviate and give rise to tissue masses called tumors or neoplasms. If the cells of a tumor can invade and disrupt surrounding tissue, the tumor is said to be malignant and is identified as cancer. And there's one more type of cancer, which is benign cancer. The properties of cancer is immortalization. The cellular machine mainly Responsible for extending telomeres is the protein telomerase, which is usually shut off but turned on cancer. Contact inhibition, it is a process of arresting cell growth when cells come in contact with each other. As a result, cells stop proliferating when they form a monolayer in culture dish. But cancer cell does not show contact inhibition. And then there's a lower serum requirement. There are many other properties of cancer cells as well. Cancers are genetic diseases. It can be hereditary. There is a high incidence of particular cancers in some human families. Descendants of cancer cells are all cancerous. It is a it is the clonal descendant of a cell that become cancer. Exposure, the incidence of cancers increases upon exposures to mutagenic agents like chemical and radiation. Cell cycle. So this is a diagrammatic representation of how a cell cycle works. So from here, there's cell cycle entry, and this is the G1 phase. Then it goes under S phase, and there's G2 phase, and then there's M phase. There are 4 stages of cell cycle. G1, the gap 1, cell growth. The G1 checkpoint ensures that everything is ready for DNA synthesis. Then DNA replication takes place in synthesis. In G2 phase, growth and preparation for mitosis. The G2 checkpoint ensures that everything is ready to enter the M phase and divide. Mitosis, cell division occurs. The metaphase checkpoint ensures that the cell is ready to complete cell division. So there are 3 major checkpoints, which is G1 to H checkpoint determines whether the cell is able to or should continue into S. The second checkpoint is G2 to M checkpoint. Unless all the DNA has replicated, the cell is big enough and the environment is cannot enter into M phase. The third checkpoint is M checkpoint. The chromosomes must be attached properly to the mitotic spindle to trigger the separation of chromatids and the completion of mitosis. So the relationship between cell and cancer is during development, a cell is produced by cell proliferation. Cell differentiation is loss of ability of cell to proliferate. They have finite lifespan in the tissue and are replaced with younger cells called stem cells that are capable of self-renewal. Both benign and malignant growth is not linked with differentiation of any tissue. That is, cell can divide without undergoing terminal differentiation. For in malignant neoplasms, example, most daughter cells from any replication event fail to express fully the genetic programs that regulate terminal differentiation. These are my references. Thank you. Sir, the malignant tumor spreads, invades other tissues, and the benign tumor only affects one particular system or organ. I don't know, sir. Uh, Sir, there won't be any regulation for cell cycle, and there might be enzymes and all एक proper order में नहीं काम करेंगे. Finally, the cell might not fully get replicated or divided? Uncontrolled रहते हैं. So yes, the cell will turn into a cancer cell. Thank you, sir.
Yes, sir. Then I'm sharing it. Hello, sir. Good evening, sir. My topic is plasmid. The detailed explanation of one of the plasmid, and the detailed explanation is on M13 plasmid and properties of one plasmid, that is lambda phage. I'm going to see. What are plasmid? The type of DNA-based cloning vector derived from a phage and plasmid. Plasmids are capable of carrying an amount of foreign DNA comparable to a plasmid, but have some special features such as ability to produce single-stranded DNA. N13 phage is a bacterial virus composed of single-stranded DNA encapsulated with a major 9 and minor 3, 6, 7 and 9 code proteins. It is long, rod-like shape, 800 nanometer in length and 6.6 nanometer in width. The M13 phage infects and grows only in the male strand that is displaying a pili of Escherichia coli. As with other lysogenic phages, M13 phage host infects the host by injecting its genetic material. The genetic material the phage the cellular host metabolism in order to replicate itself and associate protein. Those protein products are transported to the host cell membrane where new phages are packed and released through a protein pore channel in a bacterial membrane without disrupting the cell wall. Bacterial growth continues even after the phage infection, providing an advantage for mass amplification of the phages. M13 phage, it is a non-enveloped rod of filaments of 7 nanometer in diameter, 700 to 200 nanometer in length, helical capsid with absorption protein on one end. So genome, circular single-stranded genome, positive sense of 4.5 to 8 kb encoding for 4 to 10 proteins. Replication occur via single-stranded DNA, intermediate and rolling circle. Replication, cytoplasmic replication. The viral G3 protein is mediates the pilus-mediated absorption of the virus onto the host cell. Pilus recognition pulls the virion to the host in internal membrane and allow and subsequent interaction of G3P with the internal membrane protein. which is a co-receptor. The proteins of the capsid perform the injection of the viral DNA through the bacterial membrane into the cell cytoplasm. The host polymerase converts the positive sense single-stranded DNA viral genome into the covalently closed double-stranded DNA called replicative form of DNA or rfDNA. Double-stranded DNA transcription by the host RNA polymerase give rise to the viral mRNAs. The viral G2P proteins mix the rfDNA strand at the origin of replication. Positive sense strand replication occur via rolling circle. The new positive sense single-stranded DNA genomes are converted into the new RF molecules, and further transcription occurs when enhanced G5P protein is synthesized. Conversion of RF double-stranded DNA is inhibited as new synthesized genomic single-stranded DNA is converted with G5P, and G5P protein are replaced by G8P proteins to trigger the assembly of the viral capsid. New virions are secreted from the host cell. Infected cells continue to divide and produce virions indefinitely. Properties of lambda phage. Lambda phage is a non-contractile tail phage, meaning during an infection event it cannot force its DNA through a bacterial cell membrane. It must use an existing pathway to invade the host cell, having an involve the tip of its tail to interact with the specific pore to allow the entry of its DNA to the host. The phage is a phage template bacterial phage that infects E. coli. A phage is a virus particle consisting of a head containing 48,514 base pairs of double-stranded linear DNA as its genetic material, and a tail that can have tail fibers. A phage particle injects its DNA into the host through a tail, and phage will then usually enter the lytic path. These are my references. Thank you. Yes, sir. 5 minutes over. Plasmid can infect the bacteria and replicate indefinitely. That will be the advantage. And we must, no, sir. I'm sorry. Thank you, sir. Sir.